High altitudes have a considerable effect on the body and oxygen intake. The content of oxygen in the blood is determined by the percentage of oxygen saturation of hemoglobin. At around 2100 meters above sea level, the saturation of oxygenated hemoglobin in the body begins to rapidly decrease, whereas altitudes of over 8000 meters above sea level are considered to be a death zone, where the human body cannot adapt and acclimatize to the harsh environment. At higher altitudes, the atmospheric pressure is lower than at sea level, which means that less oxygen will be able to enter our blood plasma through our lungs and ultimately our tissues. In order to increase the amount of oxygen that is delivered to our tissues, the body responds in multiple ways. The immediate response from our body is increasing the rate of breathing or hyperventilation, which can be dangerous and is only efficient for a short period of time. Therefore, a much safer and long-term efficient way of delivering more oxygen to our tissues is increasing the concentration of 2,3-BPG in the blood, while also increasing the number of hemoglobin molecules, ultimately increasing the red blood cell count in the blood. This mechanism increases oxygen release from the red blood cells under circumstances where it is needed most. This release is potentiated by the Bohr effect, which we will talk about more later. The interaction with 2,3-biphosphoglycerate is considered a form of heterotropic allosteric modulation. A well-known characteristic of 2,3-BPG is to greatly reduce the affinity of hemoglobin to oxygen. BPG regulates the oxygen binding affinity of hemoglobin in relation to the oxygen pressure in the lungs and is one of the most important factors in the physiological adaptation to the pressure at higher altitudes. Under normal circumstances, approximately 40% of the maximum oxygen carried by blood is distributed to tissues, which is the result of regulation. However, at high altitudes, or 4,500 meters, the pressure of oxygen is considerably lower, which then results in the delivery of oxygen to tissues being greatly reduced. With time, the body adapts and the concentration of BPG in the blood rises, reducing the affinity of hemoglobin for oxygen. This rise in BPG concentration levels has less of an effect on the binding of oxygen as it does on the release of oxygen in tissues to restore this initial 40%. The binding site of BPG to hemoglobin is located in the cavity between the beta subunits in the tense state. BPG lowers the hemoglobin's affinity by stabilizing the tense state, and contrarily to oxygen, there is only one molecule of BPG bound to each hemoglobin tetramer. The transition to the relaxed state inhibits BPG binding. This is a result of the R state having oxygen bound to a heme group which has a different conformation that does not allow this stabilizing interaction. The binding of oxygen at each of the 4 heme irons in the hemoglobin tetramer exhibits a positive cooperativity, meaning that oxygen binding at one site increases the oxygen binding affinity at each remaining site. Likewise, oxygen unloading at one site decreases the oxygen binding affinity at the remaining sites. This cooperativity among the 4 globin subunits of each hemoglobin molecule enhances the efficiency of oxygen loading and unloading. The cooperativity of oxygen binding results from the fact that the binding of oxygen to the heme iron of a given subunit produces a localized change in tertiary structure that is transmitted to adjacent subunits, thereby triggering the shift in quaternary structure. This oxygenation-linked shift in quaternary structure between the T and R states is central to the allosteric function of hemoglobin as an oxygen transport molecule. The Bohr effect hinges around allosteric interactions between the hemes of the hemoglobin tetramer. When oxygen concentration levels are high, as in the lungs, the relaxed state is favored, enabling the maximum amount of oxygen to be bound to the hemes. In the capillaries, where oxygen concentration levels are lower, the tense state is favored, in order to facilitate the delivery of oxygen to the tissues. The Bohr effect is dependent on this allostery, as increases in carbon dioxide and H+ help stabilize the T state and ensure greater oxygen delivery to muscles during periods of elevated cellular respiration. It's also important to note that 2,3-BPG has no effect on myoglobin and that it does not exhibit the Bohr effect. The binding process for BPG, oxygen, and hemoglobin can be summarized with the following formula. Coming back to the Bohr effect, we can observe that hemoglobin's oxygen binding affinity is inversely related to the acidity and concentration of carbon dioxide. An increase of carbon dioxide results in a decrease of blood pH, which then stimulates hemoglobin proteins to release their oxygen. Inversely, a decrease of carbon dioxide brings forth an increase in pH, which then results in hemoglobin picking up more oxygen. As we all know, carbon dioxide is the main byproduct of cellular metabolism. However, carbon dioxide is not the only molecule that can trigger the Bohr effect. If muscle cells aren't receiving enough oxygen for cellular respiration, they resort to lactic acid fermentation, which releases lactic acid as a byproduct. This also increases the acidity of the blood. Like oxygen, carbon dioxide is a non-polar molecule, which means that it does not dissolve in the blood. In order to increase carbon dioxide solubility, an enzyme called carbonic anhydrase converts the carbon dioxide in the presence of water into carbonic acid, H2CO3, which is a weak acid and will dissociate into bicarbonate and hydrogen, which are both soluble in water. Hydrogen and HCO3 ions bind to hemoglobin at special allosteric sites and induce conformational changes. They decrease the ability of hemoglobin to bind to oxygen. The Bohr effect essentially increases the efficiency of oxygen transportation in the blood. After hemoglobin binds to oxygen in the lungs, due to the high oxygen concentrations, the Bohr effect facilitates its release in the tissues, particularly tissues in most need of oxygen. When a tissue's metabolic rate increases, so does its carbon dioxide waste production. As the concentration of hydrogen ions inside the blood increases, the pH of the blood decreases, which results in the blood being more acidic, which in turn affects the hemoglobin's ability to bind oxygen.